This is podcast 25. All right, we were talking about mutations in the last podcast and how that most mutations get repaired. There are many DNA repair mechanisms, and we're going to go over several here. They are absolutely essential to keeping a cell healthy. Mutations are super common, so just one type of mutation, deaminations, which happen from spontaneous hydrolysis, uh, interaction with water in the cell. That can happen up to 100 times a day per cell. So obviously there has to be something to counteract that, and there is. There are repair mechanisms, several different kinds of repair mechanisms. This is a single excision repair mechanism where you have a damaged base here, and there'll be a DNA glycosylase removes the modified base, leaving an AP site. So apurinic, missing a purine site. That's what AP stands for. Okay, so first that is cut out. Okay, the base you can see here. Here's the base, and it's missing there, and that modified base, say it's deaminated, is cut out and now just left hanging. Then there's an AP endonuclease, which is going to cut the AP deoxyribose. Okay, so the sugar that was attached to the base gets cut out. Okay, now you just have a missing nucleotide in there. DNA polymerase is going to read this strand and synthesize a new base, reading the opposite strand as a template like it normally does. Okay, and then you have DNA ligase that's going to connect this new uh, missing base that has just been added to the rest of the chain. And so this was a C as because the base pairs with a G on the opposite side. DNA polymerase adds a C there, and then ligase makes it all connect. Okay, so that would be single based excision repair by this mechanism. Okay, remember we talked about a type of mutation called thymine dimers, where thymines are absorb UV radiation, forms a covalent bond between adjacent thymines on one strand, and then they can't base pair with another strand. So there is a way to fix this in some eukaryotes and most bacteria called photolyase that uses light energy to break this bond. Most eukaryotes aren't doing that. Most eukaryotes would use a way to repair thymine dimers. So this thymine dimer repair is going to, this is another type of nucleotide excision, like we just saw. And this is one that's been worked out in bacteria. I don't know if it's known whether or not it works the same way in eukaryotes or not. But you have a thymine dimer on one of your two strands, this dark blue strand here in this double helix. Okay, the proteins called UVR A and B binds to the opposite strand, opposite the thymine dimer, and then the A proteins of this UVR AB complex leave, and a C protein of UVR complex comes in, and it'll make a 3' cut, 3' of where the thymine dimer is. Then B and C will travel toward the 5' end and make a 5' cut. So now you have on this blue strand with the thymine dimer, you have a cut downstream, 3' of that thymine dimer, and now you have a cut upstream of that thymine dimer. Now UVRD binds and helps release this piece that you have just cut out. Okay, here's the 3' cut, here's the 5' cut. D is going to bind to this now single-stranded piece of DNA and remove it. DNA polymerase gets recruited in, it does its thing, it uses one strand as a template and synthesizes the other strand according to what the nucleotides are called for on this light blue template, and then DNA ligase joins the newly synthesized piece to the old piece, and the repair is made. Okay, so this is another type of nucleotide excision repair where you're chopping out a bigger piece of DNA, not just one nucleotide base, as in the deamidation that we just saw. There are people that don't have this repair mechanism functional. They are called xeroderma pigmentosum patients. The disease is called xeroderma pigmentosum. They are particularly susceptible to skin cancer caused by thymine dimers that can't be repaired. So they would be missing any part of this UVR repair complex, A, B, C, D, or more than one of those. So they accumulate thymine dimers, but they can't repair them. Okay, here's an H5P question for you. You can have other sorts of breaks that need to be fixed. So there is a system called non-homologous end joining that fixes double-stranded breaks. So double-stranded breaks are particularly dangerous for your DNA because then there's no other side to use as a template to say, okay, what's the sequence supposed to be here? So let's say something happens where you have a double-stranded break. It's usually a more traumatic event, like a high-energy X-ray or some kind of oxidative damage, and you break both sides of your DNA. There are these donut-shaped proteins called Ku80 and Ku70 that bind to each end of the break. They will then trim the ends back because it's usually not the case that you have a break in exactly the same spot on one strand and another strand. And if that's the case, you're going to end up with a sticky end where one end has got some unbase paired nucleotides because it was cut unevenly compared to the other end. So by trimming it back, you can ensure that you'll get back to the point where it is truly double-stranded on both this side and on that side. Of course, the disadvantage of that is you lose whatever you cut back. And then you have DNA ligase that puts the two trimmed pieces back together. So after this trimming, this DNA is lost, and this is why it is such a dangerous type of mutation, is because you're going to lose DNA and you're not going to replace that DNA. The CRISPR system, if you've heard about that, as a way of causing DNA mutations, causing gene knockouts, it takes advantage of non-homologous end joining in that CRISPR will induce a double-stranded break at a specific spot of the researcher's choosing or of the researcher's design, and then non-homologous end joining comes in, and the more often than not that results in a knockout of the gene because there has been information lost, and so the gene can't be correctly transcribed after it goes through this process. If the mutation happens after the S phase, so after the S phase in the cell cycle, DNA has been duplicated. S stands for synthesis. That means that there are sister chromatids for all the chromosomes, and sister chromatids have exactly the same sequence. So it's two copies of the chromosome joined by one centromere, so it's still considered one chromosome, but the two copies in the sister chromatids are exactly the same sequence. And this synthesis-dependent strand annealing, SDSA, takes advantage of this sister chromosome as a template to fix a double-stranded break. So in non-homologous end joining, you would just trim back these ends and then put them back together, and you would lose some sequence. In synthesis-dependent strand annealing, if you have sister chromatid next to you that is the same sequence, you can trim back part of your sequence, just one of the two strands, where the break happened, so you end up with some single-stranded DNA, and then that single-stranded DNA will invade the other double-stranded piece on the sister chromatid, and it'll base pair with it, and it'll dislodge whatever was base paired in that double-stranded piece, and this is called the D loop. So the strand here in tan invades this double-stranded piece of DNA, and it dislodges this blue piece of DNA when it base pairs with it. This can migrate as the DNA moves, and it can base pair with this piece of DNA, and then base pair, and then move back, disengage, and re-base pair with the single-stranded piece on the other side of what got trimmed back. So now you have a piece of DNA that was synthesized because the DNA loop got dislodged, and then DNA polymerase came in and synthesized a new piece of DNA. That new piece of DNA can now go in here. It's kind of confusing because of the same colors, but you see here this becomes that, this here becomes that, and then this here is the new piece of DNA that was synthesized after invasion. Now that new piece of DNA that's synthesized after invasion snaps back to its original strand, and now there's enough of DNA here that it's going to overlap and cross this original break. Okay, so now that it's crossed this original break, now this strand has a template for the whole thing. So for these two here that are missing, they act as a template to fill in these two. And for all of these that are missing, this can act as a template to fill in all of that. So DNA polymerase fills in all those spots, and then DNA ligase puts them all back together. So this is synthesis-dependent strand annealing. Let me go over it one more time. So this is another way to fix a double-stranded break, which are particularly dangerous. In non-homologous end joining, you're going to lose some DNA. In this way, you would not lose any DNA. So you have a double-stranded break, and this is only if you are in a chromosome that has a sister chromatid. So it's after the S phase. This would be one chromatid, and this would be the other chromatid in one chromosome. One chromatid has a break. It's trimmed back. The single-stranded piece after that results after being trimmed back invades the other chromatid and base pairs with it. Then a new piece of DNA is synthesized going upstream from that piece. This is where this diagram is not great, and it shows that new piece being synthesized right here. Okay, follow my arrow right there. That's the new piece being synthesized. That new piece that got synthesized is going to snap back to its original piece, and now there's enough overlap that some of it will form a double-stranded piece with the that crosses the gap right there. And now you have a piece that is at least single-stranded and one contiguous piece all the way through. DNA polymerase can fix all of the bases that don't have base pairs. So these two don't have a base pair. These 1, 2, 3, 4, 5, 6, 7 don't have a base pair. DNA polymerase is going to go in and use this as a template to fill in that. Use this as a template to fill in that. It's all filled in. And then DNA ligase is going to put them all back together. That's synthesis-dependent strand annealing. Another kind uses these holiday junctions, more complex arrangement of DNA. So it starts off the same way. You have a double-stranded break. You have to have a sister chromatid. So this is after the S phase. You trim back so that part of the DNA will be single-stranded after the double-stranded break. Those pieces are going to invade from the strand that had the break and from the strand that doesn't have the break. So the part that does not have the break invades the strand that does, and the strand that does have the break invades the strand that does not. Then you have DNA synthesis on both strands. So the part that did not have the break invading in acts as a template to make new DNA. And for the part that invades in, that acts as a template to make new DNA. After this, you have these holiday junctions. So in that you have one strand crossing over into another strand and then maybe even crossing back. And in order to get two double-stranded pieces of DNA, which is where we started, you're going to have to break those holiday junctions, they're called, and you can break them at several spots. You could break them here, you could break them here, and etc. And depending on how you break them and where you break them, you'll get different proportions of original and replicated DNA in the resulting sequences. So you could end up with a situation where the non-mutated piece of DNA looks the same as it did when you started. Okay, but you could also end up in a situation where the non-mutated DNA now has crossed over DNA from the sister chromatid that had the mutation. Okay, so this is another method of fixing a double-stranded break. So we talked about non-homologous end joining, talked about synthesis-dependent strand annealing, and then we talked about this one here, which is a double holiday junction formation. Here's another way from another book that talks about this double holiday formation, and it just adds in these proteins that are important for forming these holiday junctions called REC-A. Those will be important in another class because REC-A is a mutation that shows up in cancer a bunch, and then how these are resolved in this case. You know, where after you get double-stranded DNA formed, how is it resolved at the end? So this is just another look, another sort of diagram at that. So it's one important thing about all these DNA repair mechanisms is that DNA repair is not rare. It is common because mutations are not rare. Mutations happen all the time. Mutations that are not fixed are rare. So mutations happen all the time, and your DNA repair mechanisms fix them all the time. If you have a mutation in a DNA repair mechanism, then cancer can develop really quickly, and some of the most potent cancer-causing genes, oncogenes, are mutations in DNA repair mechanisms, because, again, mutations happen all the time, and if you can't fix them, then it's likely that you'll accumulate a mutation that will lead to uncontrolled cell growth or cancer.